Well, good morning, everyone, or not morning, afternoon, everyone, and welcome to the Invasive Mussel Collaborative webinar series. I'm really glad to have our two presenters joining us today. I think this is going to be a really informative webinar, so thanks for all of those of you who are tuning in today. Our two presenters, Matt Barbour with the USGS Upper Midwest Environmental Science Center and Angelique Dahlberg with the University of Minnesota, are going to be sharing some findings, lessons learned, and alternative management strategies for invasive dry-seeded mussels. My name is Sam Tank, and I'm joined today by my colleague Patrick Caniff, and we are both coming to you from the Great Lakes Commission. And on behalf of the Invasive Mussel Collaborative, we want to say thank you for joining today. Before I turn the presentation over to our presenters, I did want to provide some background on the Invasive Mussel Collaborative for those of you who are joining us that might be unfamiliar. The collaborative convened in 2015 to identify opportunities to advance scientifically sound technology for management and control of invasive dry-seeded mussels in order to produce measurable ecological and economic benefits. The Invasive Mussel Collaborative published a strategy to advance management of zebra and quagga mussels in 2018, and that's available on our website if you'd like to read the strategy in full. The strategy includes 5 goals and a series of sub-objectives. So to make progress on these goals that are outlined in the strategy, the collaborative has established 4 work groups: the research work group, Dreissenid toxicity testing work group, the coastal site priorities work group, and most recently, the planning and implementation work group. So I just want to briefly talk about each of these and introduce some of the products that we've recently developed with our work groups. So starting off with the research work group, this group is charged with coordinating existing research and developing targeted research plans and projects that address Great Lakes management priorities. The work group recently released a coordination tool to help protect the Great Lakes Basin from the impacts of invasive mussels through this tool which allows for researchers and managers to share their work and collaborate with others to help us protect the waterways of the Great Lakes and beyond. You can see that this map is in Great Lakes focus, so anyone doing dry-seeded mussel work, whether it's applied control or research, is able to contribute. Currently, we have 120 past and current projects from across North America that are being featured. And you can find this on our website. The next product that I want to talk about comes from our toxicity testing work group, and they were charged with reviewing how toxicity testing of potential control methods on mussels has been conducted across various labs, and then discuss the strengths and weaknesses of the different approaches and methods to identify and make recommendations of what could be further refined in terms of methodology, and then standardize and develop a standard guidance to be used by researchers. So this has been a lot of work over the past couple of years, and one of our presenters, Matt, is actually part of this work group, actually Angelique too. So we're happy to have a lot of expertise participating in this group. The studies were recently compiled into this table that you can see on the screen and provide a searchable collection of literature reviewed for Dreissenid laboratory toxicity testing and open water control tests. And a manuscript has been drafted and is going for being submitted for publication likely the beginning of next month. Okay, and so one of our most recent products or tools that we've developed is led by the Coastal Site Priorities Work Group. They were charged with reviewing information on where Dreissenids are impacting Great Lakes resources and evaluating sites based on this information and other criteria, and then developing and implementing a system that allows us to prioritize sites for potential management activities of Dreissenids. So in December of last year, so just about a month ago, the work group announced the release of an invasive or interactive dashboard and map that you can see here that displays nearshore data from the Coastal Site Priorities Index that the work group developed. The dashboard and associated indices are intended to inform future applied Dreissenid research and management activities by aiding with site prioritization and selection. So go ahead and visit this tool on our website. Click around. It has a lot of really great information and a very comprehensive set of narrative to go along with it for utilizing the tool. And then our newest work group is the Planning and Implementation Work Group, which has been charged with developing a research management roadmap for site and regional scale mussel control tools and identifying stakeholders and funding priorities and remaining knowledge gaps to provide future to guide future Dreissenid control activities. So be on the lookout in the next couple of months for some exciting opportunities with that group as well. Today's webinar is part of an invasive mussel communication network facilitated by the collaborative. The purpose of these webinars is to facilitate learning and information sharing on topics of common interest within the invasive mussel community. A portion of this webinar is going to be recorded and archived along with past webinars on our website. Matt has really graciously agreed to present us some of his preliminary findings and has provided us a written overview of the presentation in lieu of the recording. However, Angelique's portion of the webinar will be recorded and available following the webinar. If you'd like to learn more about the collaborative or you're interested in staying in touch with our communication network, I'd encourage you to visit our website, which you can see as invasivemusselcollaborative.net, or join our listserv. While we always try to do our best to minimize any technological issues during these webinars, I apologize in advance if any should arise. But before we get to our presenters, I just want to cover a few logistical items to ensure things run smoothly. The audio for this webinar is automatically streamed through your computer speakers, but you do have the option to call in using the connection information that was sent in your registration confirmation email. In order to minimize any sort of potential interruptions and background noise, we have automatically muted all attendees, and attendees will remain muted with webcams disabled throughout the webinar. We do encourage questions in written format, so all attendees have access to the chat feature. So if you have a question at any time, you can go ahead and type it into the chat box that's signified by the chat bubble icon at the top of the webinar screen. Questions can be submitted anytime. We are going to listen to both presentations prior to our question period, but there will be a facilitated Q&A at the end. If you are experiencing any technical difficulties, you can go ahead and send a direct message through the chat box to Patrick, and he can hopefully help you resolve that.
Okay, thank you. Awesome. So as Sam said, my name is Angelique Dahlberg. I'm currently with the University of Minnesota working on my PhD. And today I will talk about past Dreissenid mussel control projects and some lessons that those projects have to offer. This material is currently in review as a manuscript, and I would like to acknowledge my collaborators and co-authors listed here. I would also like to acknowledge and thank our funders listed on this slide, as well as all project managers who shared and contributed data. So this crowd is very familiar. I won't really get into the background other than to say that I'll be talking about both zebra and quagga mussels today. And I'll be referring to them just collectively as Dreissenid mussels. And to set the stage, I will also say that many water body managers currently very actively work to contain and control these species. And Matt touched on this too. Currently, prevention is one of the primary strategies for controlling Dreissenid mussels in open water bodies. But there have been increasing efforts to control these species once they are introduced into a water body, such as those projects that that Matt talked about in Lake Minnetonka. And it's those control projects that I will be talking about. So Dreissenid mussel control efforts in open water have fallen into these three broad categories: physical, biological, and chemical tactics. Physical efforts have included manual removal, such as by divers, use of benthic mats to create anoxic conditions, desiccation from water body drawdowns, especially if there's a dam. Biological strategies have investigated the effectiveness of fish, crayfish, parasites and microbes on reducing mussel populations by predation or infection. And chemical efforts have used either potassium chloride or copper-based pesticides. And throughout this talk, I will refer to pesticides and or molluscicides. I'll probably mostly call them pesticides, but when I say that, I will be referring to both biological and chemical pesticides or products. We've been using these types of pesticides or products for almost 20 years. And, you know, over these 2 decades, we've learned quite a bit. Our methods and degrees of reporting have varied greatly. There are some published reports of different projects, including the ones on this slide, and those have great insight into site-specific strategies and outcomes. However, unless we look at these projects collectively, it's hard to identify patterns that can inform best practices and help guide us in future management. So we conducted a literature search and just general all angles covered search and meta-analysis of pesticide-based open water Dreissenid mussel control projects that have occurred throughout North America. Our goals were to compile and organize data from all past open water Dreissenid mussel control projects and to identify important components of both successful and unsuccessful projects, as well as knowledge gaps and themes between those projects that can inform adaptive and future management. To achieve those goals, we conducted an exhaustive review of the published literature, and we reached out to natural resource professionals. We contacted different resource managers and researchers within the aquatic invasive species community, including the Invasive Mussel Collaborative. So it's likely that you got at least one email from me in the past year. We contacted and worked with staff from control product vendors, staff within agencies who are known to have conducted control projects, and anyone else as identified by those initial contacts. And in our search and our conversations, we requested all available information on each project from the project manager, including things like summary reports, raw data, personal communication, maps. I got photos, I got handwritten notes, anything and everything that would give us some understanding of the project. For each project, we requested and organized data into pre-treatment, treatment, and post-treatment practices and outcomes. Pre- and post-treatment data included primarily survey type information, such as the total number, the location and extent of, and the methodology of surveys. There was other content that was also included, but most of it was survey related. The treatment data included information such as the product that was used, the treatment date and duration, treatment size, treatment depth, any water chemistry metrics that were taken, and any impacts on non-target organisms that were monitored, among other things. We then categorized each project's goal as either rapid response eradication, established population eradication, suppression, or research. Rapid response eradication projects were what you typically think of with rapid response projects. They were treatments of newly discovered infestations. They were partial lake treatments conducted at the site of infestation discovery, typically within about 6 months of that discovery. Established population eradication projects, sort of in contrast to that, were eradication attempts of populations that had existed in that water body for some time and that had ongoing active reproduction. And these treatments tended to treat the entire shoreline or the entire surface area of a water body. So in general, they were a little larger. Suppression projects then worked to reduce but not eliminate or eradicate Dreissenid mussels from within the treated water body. And research projects in this context included treatments that sort of explicitly tested different methods or approaches, often with representative control sites. And I do want to acknowledge that some projects fit multiple definitions and goals, but we tried to select just one goal for each project and pick the goal that best fits. To evaluate project outcomes, we reviewed all of that data that we had collected to determine if Dreissenid mussels were present within the treatment area in the short term. So on this slide, I have 4 different scenarios. This one would be if we're looking at within the treatment area in the short term, it's on the top left. So you can imagine that hatched area might be the treated area. We looked to see if they were still present after treatment within the treated area in the short term, within the treated area in the long term, outside of the treated area in the short term, and outside of the treated area in the long term. In projects where the treated area was the entire shoreline or entire surface area of the water body, we considered all observations to be inside of the treated area, and just decided that given that definition, there were no observations for outside of the treated area. And then short term was defined as the period between the first treatment between the treatment, there was no first, between the treatment and the first post-treatment observation or monitoring event, or within 1 calendar year, sort of whichever came first. And then the long term was considered anything beyond that short term. In total, we identified 33 open water Dreissenid mussel control projects in 23 lakes across North America between 2004 and 2021. We collected, as I mentioned, sort of extensive information on each project. And these data will be available in multiple formats, including, I said this was part of a manuscript that's under review right now. It'll be available in the manuscript. It will be available in the University of Minnesota's data repository. And we will also have an Esri ArcGIS Story Maps web application. So we wanted and still want to make these data available in multiple ways to reach as many audiences as possible. And now for what we found, the interesting stuff. So we just quickly, some basic background. What we found was that since 2004, there has been a slight increase in the number of annual Dreissenid mussel control projects. Shown here are the total number of projects conducted on an annual basis. So just a slight trend. We found that the size of the treated areas relative to the total surface area of the lake has not changed for rapid response eradication projects. So those have always been relatively small, and they continue to be relatively small, again, at that site of infestation. And we've seen a decrease in relative size of established population eradication projects. And what that reflects is some of those early treatments were entire surface applications, and more recently, it's been more common to do entire shoreline treatments, which are around half of a lake's surface area. So that's what that trend represents. The number of pre-treatment survey methods per project has not changed for either rapid response or established population eradication projects. So what that means is that more recent projects do not use more or fewer pre-treatment survey methods. We've been consistent in that regard. In addition to those annual trends, we also compared projects based on their outcomes relative to their original goals. So I'll put up our figure on the next slide, but before I do that, I just want to walk through how to read it briefly. So we separated projects out by goal and then broke them into short and long-term, inside and outside again. And to simplify our data, we distilled our data down into Dreissenid mussel presence by project in each of these categories. So for example, if 100% of rapid response, we're looking at rapid response projects, have at least some Dreissenid mussels outside the treated area in the long term. That's, if you can see my mouse, it's this bottom right quadrant that we're looking at. This box will be dark blue. And so that's reflective of the scale over here. And I'll also include the numeric value in the lower right-hand corner of each box if that's easier to read. And when we look at all of our data in this way, what we see is that rapid response eradication projects were generally successful at eradicating Dreissenid mussels within the treated area in the short term. Established population eradication projects were similarly successful in the short term within the treated area, and were a little more successful in the long-term eradication throughout the water body. We see that suppression projects reduced or eradicated Dreissenid mussels within the treatment area in the short term as intended, but we also see that the treated areas were small relative to the size of the entire lake, and that sort of as managers expected, these areas were recolonized from other areas within that lake, since the rest of the lake was still infested. And then research focused projects, really generally did not eliminate Dreissenid mussels in any of these quadrants shown, but that's because the emphasis was typically on refining application methods or on some sort of technique or application practice. So the goal was always a little different with those. Whoops. In going through these projects, we also found and identified 3 overarching lessons that we thought it worthwhile to share. And these lessons are by no means inclusive, but they do highlight some important takeaways. The first lesson that we came away with was that pre- and post-treatment survey methods should be designed to meet management objectives. In a nutshell, we suggest that managers define their treatment goal, carefully consider appropriate pre-treatment survey methods that will fully determine an infestation area, then delineate all life stages and use post-treatment survey methods that again will accurately assess both adult and juvenile mussel presence. And while this all sounds fairly straightforward, and it is in theory, we need to recall that Dreissenid detection can be difficult and imperfect. And one thing we noticed was that project managers that seem to, or potentially probably accounted for a detection uncertainty and then treated for all possible life stages as part of an entire population eradication project, were more likely to meet their management objectives. We also, sort of as part of this, recommend that project managers consider using the same survey techniques or methods pre and post treatment to better evaluate treatment effectiveness. The second lesson that we came away with from these projects was that regardless of which pre and post treatment survey methods ultimately got used in a project, meeting management goals will always at least partially depend on how well the treatment area was defined. And I mentioned detection can be difficult. So we suggest that project managers consider a perhaps generous definition of the infested area, and where appropriate, consider entire shoreline or surface area treatments. We acknowledge that this may result in treating areas that are not yet invaded, but I guess suggest that managers consider the costs of a failed project to the costs of a single time over treatment. And we saw in some cases projects that weren't successful the first time would then have multiple phases to them. So there's a lot for different scenarios to sort of weigh in on. Our third lesson is that control projects are potentially missing an opportunity to collect data that can inform safe and effective adaptive management. Water chemistry and environmental conditions can be essential for determining appropriate product application. And while all projects we reviewed measured some aspects of these conditions, in general, these metrics were somewhat limited. Additionally, these products can have unintended impacts to non-target organisms, and especially as we continue to use these types of strategies, understanding which non-target organisms are present and how they respond to these treatments can help project managers ensure that these treatments are doing more good than harm. And related to that, we encourage managers to quantify trade-offs between non-target impacts and the benefits of these control projects. Lastly, I want to let you know again that our data will be available in multiple formats, including, this is a preview, I guess, of the Esri ArcGIS Story Map web application that we will have. We'll also have the data publicly available in our manuscript, but the story map will look something like this when it's live. And when you move through it, there will be a map with all of the control projects that we have information on. And each project will have an accompanying narrative that will share in detail sort of the story behind each treatment leading up to, during, and after that treatment. And I think with that, I will turn it back over to Sam. Thank you.
Thanks, Angelique. That was wonderful. I'm just going to pop up my screen, which has contact information if anyone wants to reach out to our presenters after the fact. Their email addresses are up on the screen right now. Really great presentations today. Let's see, I'm going to just scroll back through and look through all of our questions. Okay, so it looks like there's been some pretty good conversation in the chat, but I'm just going to kind of ask questions to both of you as they pop up, if that works. As a reminder, questions can be entered into the chat feature, and I will ask them. Okay, great, there's Matt. Okay, so Matt, this first one is to you. There was some discussion about it in the chat already, but based on previous lab research, Doug says he found high mortality in water flea at 5 micrograms per liter copper sulfate. So it is quite likely the treatment impacted zooplankton populations. Did you monitor dissolved oxygen concentrations before and after treatment?
Oh, we did monitor the dissolved oxygen very closely, because one of our major concerns, since copper is used as an algicide, is that we could have had a DO crash. So we wanted to monitor that very closely, and that was one of the main parameters that Minnesota DNR demanded. They're very fisheries minded in Minnesota, so which is a good thing. But I heavily agree with the Cereodaphnia comment. We saw the same thing. We shifted from in those non-metric multi-dimensional scale plots, the pre-treatment and following years had more Cereodaphnia and Daphnia and so forth in them. Immediately following the treatment, there was a shift to more of our copepods, copepod taxons. So that definitely goes right in line with the standing body of literature on that and other observations. Does that answer the? There's something about chlorophyll A too, right?
Yeah, there was another question. So sorry, Matt, for making you repeat yourself. What were the chlorophyll A concentrations again?
So the background or the more stable readings were right around 3 micrograms per liter. That spike was around double that at approximately 6 micrograms per liter. Again, it's not a very alarming amount compared to some algal blooms, but it was something that was very counterintuitive for putting copper in the water and seeing a chlorophyll A kind of bloom like that. Especially since we were worried about basically wiping out the phytoplankton and maybe some plants, and then having a DO crash was the big concern. So it was very interesting to see that response out of the ecosystem, I thought.
There's a couple of follow-up questions to that as well. So any way to tease out the degree to which quagga uptake competes with phytoplankton uptake of the EarthTech QZ, and was BOD concentration measured?
We didn't not measure BOD or any like uptake rates, toxicokinetic or filtration dynamics. It would be interesting to see if it was like a grazing release, kind of like a predatory release that allowed the phytoplankton to blossom like that. So we essentially knock out veligers and a lot of our planktivores in the water column. It certainly could result in that, but given the nature of the data that we collected, we were more just trying to keep an eye on the community as a whole than trying to figure out any of those nuances. But that would certainly be something of interest to various communities.
Great. Okay. Is long-term monitoring of native mussels being conducted in terms of chronic impacts, reduced repo, destruction of release? I don't know what that word is, etc.
I'm glad somebody brought up native mussels because there is a huge conservation effort for our native unionids, especially given the biofouling issues and extirpation caused by zebra mussels. We haven't looked at any chronic conditions. There is quite a bit of literature out there from like ecotoxicological examinations on heavy metal influences on unionids. Within Lake Minnetonka itself, in recent years, we've had a very difficult time finding unionid populations. Not to say that they're completely gone, we just have been kind of striking out. It's an extremely big water body. So sometimes it can be like trying to find a needle in a haystack, which kind of goes into the, how do you set like a early detection, rapid response treatment area? Are we sure we got everything? It's kind of unknown. But as far as the reproductive aspects, I mean, all of that is of huge importance when considering making a management decision. What are the trade-offs to this treatment? Well, one of the trade-offs could be heavy impacts on your native species, right? Especially species of concern like unionids. So I think that reproductive success kind of examination would be warranted to really understand that. Sorry if that kind of droned on.
No, that was great. That was exactly, I think, what they were looking for. Okay, I think this is a more general question that was also responded to in the chat, but I'll read it aloud for those who can't see it. Is EarthTech QZ registered for quagga mussel or just zebra mussels? And then someone responded that EarthTech QZ is registered for both zebra and quagga mussels, as well as all nuisance mollusks pets, such as nuisance snails, Asian clams, New Zealand mud snails, etc. Anything to add to that?
David is actually like the technical expert on this particular product.
Perfect.
So if anybody has technical questions about EarthTech QZ, he is definitely the guy. He actually provided our projects with some very, very good technical guidance and some help with the actual logistics and everything surrounding the treatment itself. So a shout out to Dave.
Thanks for answering some questions in the chat. Appreciate that. Okay, let's see if we have any other questions that are coming in. We'll give people a few moments to put more in. We do have a couple folks who say, great presentation, thank you, great presentation. So yeah, really appreciate you sharing your work with us today. And I do want to mention that we will certainly highlight through either our digest or our feature it on the website, so you can link to it as well. The story map that Angelique had shared, that it's forthcoming as well, so everyone can take a look at that in the future. Okay. I'm not seeing any other questions. So their email addresses for our presenters are up on the screen. I'm sure they'd be happy to chat with you if you have any follow-up questions. And if you are listening to our recording, please see probably below for a brief summary of Matt's presentation, and I'm sure he'd be happy to chat with it about you if you missed it. Thanks again, Matt, Angelique, really appreciate you being here today.
Thank you for hosting, and thank you everybody for attending the seminar or the webinar.
Thank you.
Thanks all.